Alright, so this is lab 5.1 called Fractional Distillation. I'm not actually going to show you the entire lab because this lab is meant to take probably between 3 and 5 class periods. So I'm going to I've done a bunch of it already, and I'm really just going to kind of give you the gist behind it. The first thing that you need to recognize, and that if you're taking notes on this, is that fractional distillation is a way of separating a mixture of liquids based on boiling point. So it's not a mixture of solids, it's not a mixture of liquids and gases, or gases and liquids, I just said that, or solids and liquids. It's a mixture of two liquids. So when you're looking at this test tube, it looks like it might just be water or some kind of clear liquid. Actually, it's a mixture of two different liquids, and the purpose of the lab is to separate this mixture of liquids into the two individual liquids that it's made up of, and we're going to do that by boiling it based on its boiling point. So fractional distillation is the separation of liquids, a mixture of liquids based on boiling point. So I did a lot of this ahead of time, but what we would do is take about 25 cubic centimeters of our mixture of liquids, and we would put that here. That's what this started off as before I did the lab. And we have our thermometer, and we have our delivery tube. And before, the book calls for us to actually test the liquid initially to figure out what the two boiling points are. And so what we would find if we took this liquid and just did a boiling point graph where we take the temperature over time, we would see that there's two distinct boiling points. One of the boiling points is around 80, and the other boiling point is around 100. So that would be the original step, so that we could figure out what the boiling points are before we actually go ahead and separate the liquid. So we would take a sample of this and just boil it to dryness to figure out what the two boiling points are. So what we would find is that there's a boiling point at 80 and a boiling point at 100, which basically implies that there's two distinct liquids. Because if you remember back to chapter 3 on characteristic properties, boiling point is a characteristic property, and most liquids have distinct and separate boiling points. So the fact that we have a boiling point at 80 and a boiling point at 100 means that we've got two liquids, one that boils at 80 and one that boils at 100, since it's a characteristic property. So once we have that knowledge, then we can go ahead and actually do the separation by fractional distillation. Now, we have over here a beaker of room temperature water with 3 test tubes in it. We're going to collect the liquid that's being separated, each separate part of the liquid, in 3 test tubes as we go. And I'll kind of explain that as I explain the procedure. So we have our 25 cubic centimeters of our solution, and Bunsen burner, we would heat it up, and we'd watch the temperature go up. Right about the time that it gets to 80 degrees, we'll notice that our solution starts to boil. Now, from the very beginning, we have our delivery tube over here. So what happens is, when our solution starts to boil at 80 degrees, we know that when you boil a liquid, it turns into a gas. The gas will rise, and it will travel through the delivery tube, and we're going to collect it over here in a test tube. Now, what happens is that from the time it turns into a gas to the time it reaches the delivery tube and then ends up over here in the test tube, it's cooled down enough and it's already condensed back into a liquid. So we don't need to have a lid on this container to collect it, because it's not going to be in gas form by the time it gets over here to this test tube. Because by the time it's traveled through the delivery tube, it's already cooled off enough and turned into a liquid. So basically what we end up doing is collecting a liquid over here in this test tube. So we leave the delivery tube here in test tube A for as long as it takes, for as long as the temperature stays at 80 degrees. So again, we heat it up, we watch the temperature increase. At 80 degrees, we'll notice that it starts to boil, the gases travel through the delivery tube and cool down, and then condense and are collected in this test tube over here, okay? We keep it in cooler water just to ensure that the temperature stays cool enough for it to stay in liquid form. So we leave the delivery tube here in this first test tube the entire time that the temperature stays at 80 degrees. So it's important that we're keeping an eye on the temperature as well the whole time that we're doing this. So while it stays at 80, we're collecting the liquid from our mixture whose boiling point is 80 degrees, because only that part of the mixture is boiling off. The part of the mixture that boils at 100 isn't evaporating yet and turning into a gas because it hasn't reached its boiling point. So at 80 degrees, the only part of the mixture that's actually boiling is the part of the mixture whose boiling point is 80 degrees. So that's the part of the mixture that's going to boil off and then condense over here. Now, as we're keeping a close eye on the temperature, as soon as we start to see the temperature go up again, what that indicates is that all of our substance, all of our first substance whose boiling point is 80 has boiled off. Now the temperature can continue to go up again. So then we take the delivery tube and we move it to test tube B. And I think the book labels them 1, 2, and 3. I labeled them A, B, and C, but it's the same difference. So now we have test tube A, which contains all of the substance whose temperature, whose boiling point is 80 degrees. So this is part of our mixture that we've separated now. We've separated the part of the mixture whose boiling point is 80 degrees. We can put that over here and put a stop. Okay. Now, at this point, the temperature is going to continue to go up. The temperature rises steadily from 80 to 100 at this point. There's no plateau. So we plateaued at 80, and then we move the delivery tube to the second test tube once the temperature goes up. The temperature continues to rise steadily until we get to 100. Once the temperature gets to 100, we remove the delivery tube from test tube B, and we put the delivery tube into test tube C. And I'll explain test tube B in just a second. So now, at 100 degrees, we'll notice a second plateau. We'll notice that the temperature stays at 100 consistently. At this point, we still probably have about 10 or 12 cubic centimeters of our solution, or what was our solution, left over. And all of that should be water, or should be our substance that boils at 100 degrees, which is water. And so we're basically going to wait until almost all of it has completely boiled off. We don't want to wait until all of it's boiled off because we don't want to break the test tube, but we want to make sure that most of it's boiled off. So we leave the delivery tube here. We notice, again, the temperature stays exactly the same the whole time that the water is boiling, because that's its boiling point, and we know that when a substance boils, its temperature stays constant for a period of time. So the temperature stays constant at 100, and we leave the delivery tube over here, collecting all the water. Once it appears that we're pretty close to dryness, meaning we've evaporated or boiled off most of our solution, then we can remove the delivery tube and turn off the Bunsen burner. And now we can assess what are called our fractions. So let's talk about this for a second. Actually, before we talk about the fractions, essentially what we've done at this point is we started with a mixture of two liquids. We took the mixture, we heated it to where it boiled. We then boiled off part of the mixture, which is the mixture, the liquid that boils at 80. That's in this test tube. And we've separated it completely from the substance or the liquid that boils at 100. Okay, so these two are two different substances, one that boils at 80, one that boils at 100, that were together in solution at the beginning. So we took our solution, our mixture, we boiled it, and we separated it based on two distinct boiling points. So now we have two distinct liquids. So that's fractional distillation, and that's how that works. That's the procedures behind how fractional distillation works. Now, there's a little bit more to this lab besides just the process of separating our liquids. What they have us do is to test the properties of all of the different liquids that we've collected. Instead of me showing you how to go through and test all of the different properties, I'm going to show you a sample data table and kind of explain how this works. So let's move our camera. See if this will work for us. Okay. There we go. Let's see if we can do that. Perfect. Okay, so this is a sample data table from, actually I'm going to stand on the other side. That's going to be a little bit weird. This is a sample data table from the lab, if we had actually gone through and tested all these different things. This is why this lab takes many, many days, because there's so many different things we have to test for. So first, let's start with our solution. This is what we would have done on day one at the very beginning. We would have taken our 25 cubic centimeters of solution right here, and we would have tested it for various properties. What we would have done is we would have taken a paper towel and dipped it in the solution, passed it through a Bunsen burner that was lit, and to see if the solution would ignite, if it would go up in flames. And yes, it would. So our solution is flammable. It does have an odor, not too strong, but there's a definite odor there. If we measured a sample of the solution, its volume and mass, we'd find that its density was 0.91, or somewhere in that ballpark. We also noticed that the boiling point is not constant. And here's what that means again. We already talked about this. The boiling point would look something like this, a boiling point graph. Remember that when we heated it up, the temperature would continue to rise steadily, and then when we get to about 80 degrees, we have one plateau. That's the boiling point of the first liquid in the mixture. Once all of that boils off, the temperature goes up again until we get to the boiling point of the second liquid in the mixture, and that's at 100. So when the data table says the boiling point of our solution or our mixture is not constant, that's what that means. It means there's two different boiling points. And then if we were to test it for sugar solubility, we would find that yes, sugar does dissolve in our solution. Okay, so the reason we test all these properties at the beginning is to then compare it to the different parts that we collected during the distillation process. The word fraction, basically, you guys know what a fraction is. A fraction is just a part of the whole. So if our solution is the whole thing, fraction 1 is the first part that we collect, fraction 2 is the second part that we collect, and fraction 3 is the third part. We're going to talk about fraction 2 last. Now, fraction 1, if we did all these tests on fraction 1, these are the data we would come up with. We'd find that it is flammable. We'd find that its odor is very strong. Its density is 0.84. It boils at 80 degrees, and it does not dissolve sugar. Okay. Fraction 1 is our isopropanol, which is rubbing alcohol. So clearly rubbing alcohol is flammable. We all know, if we smelled it before, because a lot of us have this at home, that it's very strong. Its density is about 0.84. You wouldn't necessarily know that or not. We know it boils at 80 because that's the temperature at which we collected it. At 80 degrees, it boiled off, and we collected it in a test tube at that temperature. If we then took that fraction 1 that we collected, or I called it A, if we took A and boil it again, we would find that its boiling point is 80. And rubbing alcohol does not dissolve sugar at all. Now, our fraction 3 is water. At the very end, the last thing we collected was water. If we tested it for flammability, we would find that it is not flammable. So if you take a paper towel and you dip it in the water and you pass it through the Bunsen burner flame, it does not go up in flames, which is interesting because the solution is flammable, and so is fraction 1. Fraction 3, which is our water, is not flammable. It has no odor whatsoever, which in my opinion is the most fascinating part, because we have this solution up here that's has a very strong odor, and not right now. Sorry, somebody's knocking on my door. We have this solution over here which has a very strong, the solution has an odor. Fraction 1 is a very strong odor. By the time we get to fraction 3, the odor is completely gone, which I find interesting. If we measure its density, it's about 1. Its boiling point is about 100, and it does dissolve sugar. Now, here's the purpose for measuring all these properties of the different fractions. There's two purposes, really. First is to show you that we do collect two distinct liquids. We know that they're different because they have different flammabilities, different odors, different densities, different boiling points, different sugar solubility. They're different in every possible way, except for the fact that they're both clear. Every possible characteristic property is different. So we know for sure, based on this data, that the two liquids we collected at the beginning, the first one and then the last one, they have to be two different liquids. So that kind of proves to you that even though when we're looking at our original mixture, it looks like it's uniform, it looks like it's exactly the same, it could be water, it could be isopropanol, it's hard to tell what it is. And so you can't really be sure until we distill it and we separate it and then we test it and we see, wow, in fact, that was a mixture. made up of two different liquids with two completely different sets of characteristics. Now, the other thing that is interesting is that if you combine fraction 1 and 2 to make our solution, you find that a lot of the properties are somewhere in between the properties of the two parts. For example, even though water is not flammable, when you mix it with rubbing alcohol or isopropanol, it becomes flammable. It has a medium odor, which is right in the middle between no odor and a strong odor. Here's what is also interesting. Its density falls right between the densities of the two liquids. So rubbing alcohol is around 0.84, and then water is around 1. So when you mix them together, the density of the mixture falls right between the density of the two liquids. The boiling points are not constant. That does not change. You can't combine a liquid that's boils at 80 and a liquid that boils at 100 and find a liquid that boils at 90, because that would make it seem like it was an entirely different substance, and that's not the case. So we find that the boiling point's not constant when we combine two liquids with different boiling points. And then with regards to sugar solubility, isopropanol does not dissolve sugar, but water does. So when you mix them together, you have a solution that will dissolve some sugar as well. The last thing I want to mention is fraction 2. Okay? Fraction 2, let's go back to our graph real quick. If we look at our graph over here, we think about how we collected our substances. We collected the first fraction here, fraction 1, when it was boiling at 80 degrees. This was our isopropanol, or our rubbing alcohol. Fraction 2, we changed, see how it says change collecting tubes? Right as we notice the temperature start to go up again, we move the delivery tube into that second test tube. And this whole time, until we get to about 100, this whole time here, we're collecting that second fraction. Then we move it to the third test tube and collect fraction 3 here. So fraction 3 is just water. Fraction 1 is just rubbing alcohol or isopropanol. Fraction 2 turns out to be a combination of both. So maybe some of that rubbing alcohol is still kind of boiling off here, and a little bit of the water starts to boil off here. And so in between these two fractions, we get a mixture of both. So if we look at fraction 2 and we look at its properties, because fraction 2 contains a little bit of this and a little bit of that, we find that its properties are almost exactly the same as the properties of the solution, which makes sense because if you think about the solution is made up of water and rubbing alcohol, fractions 1 and 3. So if this is a combination of these two, and this is a combination of these two, then it makes sense that their properties would be the same. They're both flammable, they both have a medium odor, their densities are just about the same. We notice that if we boil fraction 2, which they would have us do in the lab, we would notice that fraction 2 would look like this. It would boil, there'd be part of it that boiled at 80 and part of it that boiled at 100, just like the solution. And then we would find that there's a slight solubility with sugar, kind of like we got with our solution. So that's what the reason we look at all of these properties is to show you that, in fact, our original solution was made up of two distinct liquids mixed together. We separated our mixture of liquids based on their boiling point, which is called fractional distillation. Then we tested the characteristics of our two distinct liquids in order to prove that, yes, in fact, they are two distinct liquids that were originally mixed together into one solution. So that is fractional distillation.